Dear Assistant Professor Gonca Erdemci Tandogan and esteemed participants, a warm welcome to Bioinfocongress VII. We are truly honored to have Assistant Professor Gonca Erdemci Tandogan with us. Dr. Erdemci Tandogan, welcome once again. I wanted to say that the following question, we will have an engaging question-answer sessions where we will have addressable questions submitted by our participants and those thoughtfully prepared by our team. And we will look forward everyone's participation to enrich the dialogue even further. And Dr. Erdemci Tandogan, if you wish, you can start your presentation.
Yeah, thank you very much. Thank you very much for the invitation. I'm very excited to be here today and sharing some of our work. I'm an assistant professor at Western University in Canada in the Department of Physics and Astronomy, and my group studies physical mechanisms behind biological processes, and currently we are more interested in cells and tissue mechanics, and that's what I'm going to be sharing today. And the plan for this talk, I will start with a brief introduction of the cells and tissue mechanics, mechanical models that we use in my group, which are mainly called the vertex models. And I will be also introducing some extension to standard vertex models. And on the second half of my talk, I will be talking about how we use these type of mathematical models and combine them with in vivo experiments, specifically in this example, a cell migration mechanism for the fruit fly heart formation. I'd like to start with introducing the idea of collective cell movements because that's the motivation behind a lot of the mathematical models that we developed. Much like sheep in flock and fish in swim school, cells move collectively in groups. And these are, of course, different length scales. Sheep is much larger than cells, of course. But overall, the collective movement emerged from simple interactions between moving individuals. And in this case, these individuals are individual cells. You can see that in this movie, these cells, individual cells, move all together and create this collective behavior, which is the tissue, global tissue, tissue material properties that we're interested in. And this collective movement of cells are so important. Collective cell movements guide embryonic development and tissue repair and can contribute to disease progression such as cancer metastases. And I'm going to show you two examples of collective cell movements. On the left here, you're going to watch collective cell movements during zebrafish embryonic development. And on the right, it's going to be collective cell movements during wound repair in a fly embryo. The cells here on the top, these polygonal shapes, these are individual cells. They move all together collectively and cover the sphere, which is the yolk of the embryo. And this bright cable that you see at the edge, it is a tension-bearing cable. So there are mechanical forces there. These pulling forces indeed pull these cells and help the migration of these cells. So there are forces involved in this type of processes, mechanical, physical forces involved in these type of processes while the embryo is developing and cells are moving collectively. And on the right here, this is these more elongated cells that you see. It is the epidermis of the fruit fly embryo, which is the skin of the fruit fly embryo. And in the middle here, you are seeing a laser cut, which creates this big wound between the cells. This gap is the wound. And you can see here again a bright cable, which is again a tension-bearing cable. This contractile tension contracts the cells and eventually helps these wound to be closed. And the cells around the wound, you can see that they are stretching. They're actually collectively migrating and moving to be able to help the closure of this wound. But you can also imagine that these cells are stretching and actually pulling the rest of the tissue together. So we can appreciate that actually the rest of the tissue material properties might be important in this process as well. And indeed, it's similar to inactive materials. Biological tissues display gas, fluid, or solid-like features. For example, they display physical material properties similar to physical matter, gas, fluid-like, solid-like features. But what is really interesting, what makes biological tissues so interesting, is that they can go through phase transitions by changing their microscopic features even slightly. So, for example, they can go from fluid to solid, a jamming transition, or solid to fluid, an unjamming transition. But why would biological tissues do this? Is this something important for their function? Is it something important for disease progression or proper embryonic development, for example? And there have been recent studies suggest that this could be important. For example, in this study, they study asthma cell lines, and they show that tissue fluidity might contribute to asthma disease progress. Or in the study, they study cancer cells, and they show that cell shape and tissue fluidity correlation between the cell shape and tissue fluidity could be a novel way to predict cancer metastasis risk. And in the study, they show that there's a jamming transition happening while a zebrafish form its body axis, while a zebrafish developing and going through the body axis elongation morphogenetic process. So these are exciting studies in the last 10 years or so, and they support this exciting hypothesis that phase transitions could be important for disease and proper embryonic development. But then the second question is, if these are important, how do we control the transition so that maybe we can control the disease progression or proper embryonic development? And these are the two fundamental questions. Our modeling, mathematical modeling, the models that we develop try to address in collaboration with experimentalists and performing experiments. And answering these questions, again, mathematical models play a key role. And the standard mathematical model that we use in my group is called vertex models. Here is an example zebrafish tissue, and here's an example vertex model simulations represent this tissue in 2D. Each cell here represented as a polygon. So these individual cells represented are as polygons. These are called cell shape-based models. But cells are, of course, in 3D. But most of the molecular factors that contribute to cell shapes are distributed on this top surface of these monolayers, the apical surface, and these 2D vertex models model this top surface. There are adhesive sticky molecules, I call them sticky molecules, the adherence junctions between the cell-cell interfaces. These sticky molecules help cells to stick together, and it creates an adhesive tension between cell-cell interfaces, and cells would like to increase that interface. And at the same time, there is active cytoskeleton of the cells, which goes around the cell membrane. And this creates a cortical tension, which tends to shrink the cell area. So there's a balance and force balance between this adhesive tension and the cortical tension around the cell membrane, which gives rise to the final shape of these cells. And in this type of models, we write a mechanical energy. These are mechanical models. This is the total mechanical energy of the tissue in terms of cell geometries, cell area, and cell perimeter. These are actually very simple models in a sense that this is nothing but a spring energy. So if you consider, for example, a 1D spring, the energy of a 1D spring is 1/2 k delta x squared. And similar to that, here we have k delta a squared and k delta p squared. Ka here is the spring moduli of an area. AI is an area of a cell, and A0 is the preferred area of a cell. So the area that the cells would like to achieve, the rest area. Kp is the perimeter moduli, and then Pi is the perimeter of a cell, and P0 is the preferred perimeter of a cell. Again, cells would like to achieve a certain shape, and any deviations from this shape cause an energy penalty, and that is this energy penalty for our system, the total energy of the tissue. And from here, in this type of simulations, we write down this energy functional, and then we write down our equation of motion, which is this differential equation. In this case, Rk represents the vertex of a polygon in these simulations. This is the position of a vertex, and this vertex evolve over time. We move this vertex based on this equation of motion, evolve over time based on the forces acting on this vertex. This is the inverse friction, and this is the force acting on a vertex of a polygon, of a cell. And this is nothing but the negative gradient of the energy functional. And finally, this last term is a Gaussian noise that we introduce because these are biological systems. There are stochastic fluctuations, and that represents, this term represents those stochastic fluctuations. And you can appreciate from this movie that the cells are jiggling, and that jiggling piece that we do introduce some stochastic fluctuations there. It's interesting, these models, they are quite simple physical models, but when these models first discovered a transition point, it was actually quite a big deal. It is a very interesting feature of these models. These models have a phase transition, go to a phase transition, exhibit a rigidity transition, just like what we see in physics and physical materials. And we quantify that often from the long-term behavior from these simulations, such as calculating a shear modulus or diffusivity. If we non-dimensionalize this energy functional, we get an order parameter, and that order parameter is called target cell shape index. It's a very simple term because it's just a dimensional cell shape. It's the perimeter of a cell divided by the square root of area of a cell. And the vertex models exhibit a rigidity transition at a critical, or this order parameter, at a critical target cell shape index. And again, we calculate this rigidity transition based on the long-term behavior of these simulations, either calculating the shear modulus, for example, or diffusivity. And here's an example of diffusivity. This plot shows us diffusivity as a function of target cell shape index. And diffusivity is a measure of how diffusive particles are, which in this case, these cells in these simulations. And if the target cell shape index is larger than this critical value, the tissue, interestingly, acts as a fluid-like. If it is less than this critical value, the tissue acts as a solid-like. And here are two examples, like these simulations from these regions. In the fluid, this is how simulations look like of these cells. In the fluid, these are cell trajectories. Cells move around freely without paying energy penalties. And the cells, if you notice here, they are elongated. They're this like pancake-shaped type of cells. And in the solid, here's an example configuration from the solid region. Cells are more roundish, and these are cell trajectories. Again, the cells do not move much more than their original position. They are jammed, and that's the difference what we observe between the cell trajectories and the fluid region versus the solid region. One thing you can notice here that these are confluent tissues, so meaning that there are no gap between the cells, right? It is polygonal tiling. There is a polygon next to another polygon. And in order for these cells to be able to move around, just like in these trajectories, they need to exchange neighbors. And this is called a T1 transition. So if we zoom into 4 cells here, if we follow the edge that I highlighted in red, during the dynamics of the simulations, this edge shrinks to a critical length. And when that critical length reaches, we flip that edge and a new edge forms in the opposite direction. And such that these 2 cells were neighbors before, but after the transitions, they are not neighbors anymore. And these 2 cells were not neighbors before, and after the transition, they became neighbors. So this is called a T1 transition. This is how cells in general move in confluent environments. And in standard vertex models, in this type of simulations, T1 transitions are modeled instantaneously, meaning that at that simulation time step, while these cells are evolving, whenever this critical length is achieved, we do the flip and form a new edge, and at that time steps, instantaneously T1 transition happens. However, in biological processes, during tissue formation, during embryonic development, for example, cells rearrange in finite amount of time. That's instantaneous, very quick time is not possible, as this edge needs to be shrinking and then forming a new edge, and that takes time, quite a bit of time in real systems. So just to summarize so far what I show you about these type of standard vertex models: elongated cell shapes associated more fluid-like behavior. And these are homogeneous cells. One thing I didn't mention, but you can notice that these are all same cell types. And these are homogeneous cells, one single cell type. And in standard vertex models, again, this rearrangement is happening instantaneously, which is in real biological tissues. It happens in finite amount of time. For example, here's an example. For a biological process, this is a cartoon of a fruit fly embryo. This darker gray region that you see is called the germ band of the fruit fly embryo, which over time this extends and then the tissue becomes here. This is called embryonic body elongation. This is a very preserved control the morphogenetic process. It's true for across species in fly embryo, in zebrafish embryo too, for example. That's called embryonic body elongation. And if we zoom a region in this case to the tissue, this is how you see cells in 2D. And if you watch these trajectories of these two cells highlighted here, you will see that the tissue is flowing. That's the body elongation. It is elongating. And during this process, cells are rearranging. This is a confluent environment again. There are no gaps between the cells, and they exchange neighbors and they rearrange. And that rearrangement is actually quite a long time. It takes about 10 minutes to shrink this edge to a critical length and now form a length in the opposite direction. It takes about 10 minutes. And this whole process is about 30 minutes. So compared to the morphogenetic process here, 10 minutes is quite a bit of a long time scale. And we wanted to ask the question, we wanted to introduce this time scale into the standard vertex models and ask the question that, how does the cellular rearrangement time scale affect tissue material properties, or whether it affects tissue material properties or not? To answer this question, we introduce a timer into the standard vertex models. For example, this is a snapshot from my simulations. What you see here is an edge, and we are zooming into that edge. So there are 4 cells neighbor here. This edge shrinks to a critical length, and an opposite, and then over time we form an opposite edge in the opposite direction. And this time, from this configuration to this configuration, we call it TT1 time, the cellular rearrangement time, and we introduce a finite time scale to the simulations, and we do not let this edge to flip until this time is up, until the TT1 time, the timer is up for TT1 time. And to then study whether this time is important for the material properties of the tissue or not, we calculate characteristic relaxation time of the tissue, which we calculate from self-overlap function. Self-overlap function is a kind of a typical analysis in granular materials and on physical materials. Scientists use to quantify how much the particles move in the system based on a critical distance. For example, if the self-overlap function is 1, it means that none of the particles move in the simulations. If it is 0, it means that all of them move in the simulation. So it's a weight function. It's for each one of these particles. If they move more than a characteristic distance or not, we quantify them, and it tells us, at the end, it tells us how much of that moves in the tissue. And we specifically look at the fraction 1/e. When the self-overlap function is 1/e, the time corresponding to this point we call the characteristic relaxation time. So meaning that at this point of the simulations, most of the particles move more than a characteristic distance, which is a cell size distance. That's what we call the characteristic relaxation time. And first, we analyze the characteristic relaxation time of the tissue with TT1 time is 0, with instantaneous cellular rearrangement time as in the standard vertex models. Here's the characteristic relaxation time as a function of target cell shape index. As I mentioned in the beginning of my talk, target cell shape index is our order parameter, and there was a rigidity transition for standard vertex models with instantaneous rearrangement time. And as the predictions, as supporting the prediction of standard vertex models, this relaxation time decreases. This relaxation time decreases as the target cell shape increases, as the more elongated cells associated with more fluid-like tissue, and it takes less time for tissue to relax. So supporting the standard vertex models predictions. Now, to answer the question whether when we increase this time, whether when we have a finite time for the rearrangement time, whether that impacts the tissue material properties or not, then we calculate the characteristic relaxation time. We analyze the characteristic relaxation time as a function of the cellular rearrangement time for various cell shape values. So this plot includes a little bit of too much information. So I will describe it by step here. First, in this first region, I would like to get your attention to. In this region, here, I should have mentioned, we normalize both axes by the characteristic relaxation of time of the tissue with instantaneous rearrangement time. So in this region, if tau alpha over tau alpha 0 is equal to 1, that means that in this region, independent of the rearrangement time, tau alpha equals to tau alpha 0. So in this region, the cell shape dominates, as in the standard vertex models predictions. The tissue material properties is defined by the cell shape. If the rearrangement time is low, it's a short time, it doesn't matter, even there's a delay in the rearrangement time, still cell shape is the main factor that defines the tissue material properties. However, in the second region, the characteristic relaxation time increases significantly by rearrangement time delay. And this is actually a slope of 1. So if this is a slope of 1, this axis will be equal to this axis. So in this region, then the characteristic relaxation time, if this is a slope of 1, will be equal to TT1 time. So in this region, when we have a longer time delay, when these cells take longer time to rearrange, the relaxation time of the tissue approximately equal to this delay time that we introduce in these simulations, suggesting that independent of the cell shape, here rearrangement time dominates, and rearrangement time defines the the tissue material properties. So this actually proposed something quite interesting and exciting, in my opinion. This suggests that actually might be organisms can utilize this time scale to control the global tissue response, to control the tissue material properties regardless of the cell shape. And all I show you so far for this type of simulations with the rearrangement time was with an isotropic periodic boundary conditions and isotropic type of simulations, but many biological tissues are indeed anisotropic. For example, I would like to go back to this cartoon where I introduced embryonic body elongation. If we zoom into this tissue in this region, and this darker gray region extends over time, and this tissue is an anisotropic tissue, and here's the anisotropy here. There is a polarized tension between cell-cell edges, which in this cartoon represented by these red lines here, which is the internal stress from polarized tension, which the myosin tension-generating protein localizes and creates these cables for the tissue, which creates this anisotropy. Or there's also another source of anisotropy here. There are external stress coming from the other tissues in the embryo, and these external stress can actually shear this tissue over time and then eventually contributing as an external force to the tissue. So next, we wanted to model this. We wanted to simulate anisotropic type of tissues and then study whether this rearrangement time is important for this type of tissue, as we saw in isotropic type of tissues. I'm not going to, in the interest of time, I'm not going to go into too much details of how we introduced this, but happy to chat later or happy to answer emails. This is the energy of the total tissue again. And as I introduced in the beginning of my talk, the first two terms is actually exactly identical. It is the area spring term and the perimeter spring term. Here we introduce one more term to these simulations to create these anisotropic tissue, to create these cables in the tissue, just introducing an additional line tension based on the angles of the cell edges. And in the second case, we apply a pure shear strain. We shear the simulation box, and then this creates the box to be sheared and extend over time. And this is how our simulations look like in this case. We start out with an isotropic type of tissue. We start out as an isotropic type of tissue, and over time, this tissue extends, and we create this anisotropic box. We repeated our analysis, we repeated our calculations, and in this case, we found the same results as in the isotropic type of simulations. This is the characteristic relaxation time as a function of the rearrangement time. And we found here again one more time, if the rearrangement time is not long, if it is short, then in this case, the cell shape dominates. And in the second region here, what we observe is that this is a slope of 1 again. Characteristic relaxation time is equal to the T1 delay time we introduced in this type of simulations. And to conclude this first part of my talk, we show that increasing cellular rearrangement time can stiffen the tissue, which we characterize it from the characteristic relaxation time. And we show that if this rearrangement time is long, then the tissue becomes stiffer regardless of the cell shape. Increasing cellular rearrangement time increases the number of rosettes. In the interest of time, I did not introduce the rosettes here, but more of the story is you can find it in this paper. And the other conclusion of this result was that this model proposed that organisms might be able to utilize this rearrangement time in order to control the tissue material properties. And this also suggests that it is important to investigate which type of molecular processes define rearrangement time. And finally, rearrangement time, by studying the anisotropic tissue simulations, we actually showed also that the rearrangement time can control the body axis elongation. So it could be actually an important factor that is affecting a morphogenetic process, which is in this case, the body axis elongation of the embryo. Now I'm going to talk about, in the second part of my talk, I'm going to talk about an example of how we use these type of mathematical models and then develop hypotheses and test them in actual embryonic data in vivo. And specifically, this is a project to understand cell migration of fruit fly heart cells. Here is a cartoon of a fruit fly. And these red cells that you see at the two ends, these are called cardioblasts. These cardioblast cells migrates from two ends of this embryo and meets in the dorsal midline here form this tube. And this heart tube is as simple as it is, or as complicated as it is the heart of the fly embryo. And here is a movie of this process. You see here highlighted cell nuclei of cardioblast cells at the two ends of this embryo. And you also see some nuclei neighboring those cells. These are called pericardial cells. These pericardial cells are the neighbors of the cardioblast cells. And you will see that in this movie, they migrate over time. They come from two ends and then migrate and meet in the midline and eventually form the heart tube, which is the fly, fruit fly heart tube. And you will see that this is actually, I mean, relatively fast process because the fly embryo develops quite fast. But at the same time, you can see that there's a kind of a slow process that the cells are not directly moving to the midline. They are this like jiggling, moving back and forth, actually, which I will show in a second. We will zoom this region of this movie. And in this case, if we zoom, we will see more clearly that these cells are migrating, but they're not only migrating, they're not doing like a directed motion, but they are also doing backward, forward, and backward movements while they are moving and meeting in the midline of the embryo. You see these green lines, these are some annotations. We annotate these and then analyze this migration. Here is the position of these cells over time. And here is the velocity of these one of the cells over time. And we can see that they eventually move forward. But while they are moving forward, they also do backward motion. So they are moving forward and backward and forward and backward, but eventually most meet in the midline. So this is the cell nuclei. We also looked at a protein called myosin, which in this case, you see these highlighted regions where myosin localizes. I would like to get your attention to this region. And you will notice that not only these cells move backward and forward, they also, the myosin levels also here changing, and the myosin localizes at the front of the cell and the back of the cell. So the myosin keeps oscillating as well. And when we quantify this, when we look at the myosin behavior, we see the cyclic motion of myosin protein between the trailing edge and the leading edge of the cell. So the leading edge is the front of the cell and the trailing edge is the back of the cell. And interestingly, we see that myosin protein, there is a cyclic accumulation of the myosin protein. Since myosin is a protein, localizes at the cell membranes, and it's a protein associated creating with cell shapes, we also wanted to look at how the cells change their shape during this process. Here we are zooming in more to the cells. These green labeled edge, these are the lateral edges. And then the blue is the front of the cell, and the red is the back of the cell. And as you can notice here, we measure the cell edge length. The cell edge length also oscillating. The length keep decreasing and increasing both for the leading edge and the trailing edge. It is less for the trailing edge, but still you can notice them from the movie that it contracts, spring back, then the back contracts and spring back. So there is this oscillatory cell shape changes happening at the same time. And here's summary of what we know from the experiments, and then I will show how we integrate these into our physical models. We know that the cell nuclei oscillate back and forth, and we know that there is a cyclic myosin flow between the front and the back of the cells. And we also know that there's cyclic cell shape changes happening. The cell edge in the front and the back contracts and spring back, and contracts and spring back. So knowing these information from the experiments, we wanted to ask the question whether having such oscillatory behavior for the myosin and the cell shape changes is important for the cell migration or not, which is eventually the cell migration is crucial for the embryo. These cells migrate and form the heart of the fruit fly embryo. So this was our main question, what is the role of the cyclic myosin flow on cell migration? To help answering this question, we developed a mathematical model using vertex model framework. Again, in this case, we have 4 cardioblast cells, modeling 4 cardioblast cells, and we write a total mechanical energy for these cells. It is very similar. The first term is very similar to, I mean, it's identical to the first term that I showed in the beginning of my talk, which is the spring area term. Ka is the spring moduli and area moduli, and An is the area of a cell. A0 is the preferred area of a cell. And the second term here, we introduce one more term here, which is a spring restoring energy. As you notice from the movie is that the front and the back of the cell, it's it contracts and spring back. So it behaves like a physical spring. So we introduce a spring energy to mimic that behavior. And the last term here, we introduce an additional line tension at the front and the back of the cell, which we call leading edge and the trailing edge. And we introduce this additional line tension using a sinusoidal function, because we know from the experimental data that the edge is contracting in a cyclic fashion. So that's why we have a line tension, which gamma naught is the magnitude of this line tension. And as we use a sinusoidal function to mimic the cyclic contraction. And T here, the capital T, is the period of these oscillations, like period of how quickly we are going to contract the back and the front of the edge. You see here the sinusoidal function such that the trailing edge has 1 minus of the sine function, which there's an anti-correlation. If you notice from the movies, which I will show again, when the front contracts, the back expands, and then when the back contracts, front expands. So we put an anti-correlation between the front contraction and the back contraction. We also put in this anti-correlated behavior between the neighboring cells as well. When the front of the first cell contracts, the front of the second cell expands. So to mimic that, we have 2 types of sinusoidal function. And from here, we solve our equation of motion, meaning that we want to know this position of these vertices, where they go in time. So we solve this equation of motion. F here is the force acting on each one of these vertices, which is negative gradient of the energy functional. And here is how they look like in the simulations. If you have these 4 cells, and based on this mechanical total energy, if you solve our equation of motion and we contract the front and the back of the cell edges, it's not surprising that what we see is that the edge contracts and expands back, but these cells are remaining where they are not moving forward. So the cyclic edge contractions is just changing the cells as we would expect, that the cells change their shape in oscillatory fashion, but the cells are not moving forward. But our question was that, is this important for the cell migration? How are we going to make these cells move forward? Next, what we did in the model is that the front of the cells, they contract and expands back. The back of the cells, they contract and expands back also. But what we know from the experimental data is that the contraction expansion of the back of the cells are less. That's what the experimental data tells us. And what we also know that these are cardioblast cells, but there's also some neighboring cells which are called pericardial cells next to them. And we then propose that, what if that there's an asymmetry, a mechanical asymmetry here, such that the back cells act as a barrier, and there's a physical stiff back boundary here. When we did that, when we introduced the stiff back boundary, then the cells starting changing their shape, contracting and expanding, and started moving forward. So in this simulations and this type of migration, we do not apply any external forces. We do not pull or push these cells. They do move forward. And this is the what model process proposed for this process. The stiff boundary that we introduce in the back of these cells, plus the periodic cell shape changes, produce a forward motion for them without us pushing or pulling without an external force. And we wanted to see in the experiments if this physical boundary is there or not. So we are changing the orientation here a little bit. Now this is the cartoon of the fly embryo again. So we flip the embryo looking from a different orientation. These are the cardioblast cells. They are migrating and meeting in the midline. And this is the second group of cells, the pericardial cells that you see on 2 ends of these cells again. And actin is another tension generating protein, which localizes between these group of cells. You can see that the actin is you can see this bright cable, which is a supracellular cable. So actin localizes there and creates a supracellular cable, suggesting that there is a physical boundary between these 2 neighboring cells, between the cardioblast and pericardial cells. So going back to the model, there are many parameters involved in these simulations, of course. So lots of parameters, but I would like to get your attention to two very important ones. One is this gamma 0, which is the magnitude of the tension that we apply to these edges that helps us to contract the edge. So how much we are contracting these edges. And the other one is capital T, which is the period that is like how much the myosin will oscillate back and forth, like how quick, sorry, how fast it's gonna oscillate back and forth. So this is that we wanted to study the role of these parameters in the simulations. Here's the tension amplitude, that is how much tension we are putting to the edges, and this is the period, how fast we are putting that edges, like how quickly we are putting those tension at the edges. And the color here represents the cell displacement. And you can see that here, the darker red region is the most cell displacement. So the cells migrate most here. And in that condition, the higher contraction and the fastest, the quicker myosin oscillation leads the most cell displacement. And then we did quantify this. We did a few quantifications and compare this with the experimental data as well. This is the cell displacement over time for the free case, meaning that if there's no back boundary, if there's no asymmetry in the mechanics, so this is the cells oscillate back and forth, they do not move forward. Red is the constraint, meaning that we put a boundary and we stiff boundary and we do constrain the backward movement, and eventually the cell moves forward. The cell displacement here is about 15 micrometers. And in the reduced tension case, this is another part of the story that I'm not telling in the interest of time, but it is in this paper, which very briefly in the reduced tension case, we do laser ablation experiments and cut the back boundary, and we do repeat that same thing in the simulations, and we show that actually removing that boundary slows down the migration, and that's what this gray curve here represents. So to summarize this part, we propose a mechanism for cell migration, which is cardioblasts move by taking discrete steps, and myosin waves oscillate between the leading and trailing edges between the front of the cell and the back of the cells. And mathematical model here that we developed propose that the stiff boundary plus the periodic cell shape changes produce a forward motion without us putting any external force on these cells. And we found that there's a supracellular actin cable creates an asymmetry in movement. And overall, the conclusion is that a mechanical asymmetry can bias the direction of cell movements. In this case, it is very important for the proper formation of the heart tube of the fruit fly, which is a crucial part of the proper embryonic development. And this is just a cartoon summarizing the story that I told you, but there's more to that as we are blocking the myosin movement, myosin accumulation, and also doing laser cuts to stop the motion, and more of that is in this, you can read that is in this paper. And with that, I would like to thank people who contributed this work. The first part of the story that I told about the extension of the vertex models with the rearrangement time, I did that together with Lisa Manning, Professor Lisa Manning from Syracuse University. And you can find that story, more of it in this paper. And the second part, the cell migration, is done in the collaboration with Professor Rodrigo Fernandez Gonzalez lab from University of Toronto, and Nigar Balagi is a PhD student and who did all these experiments that I show you today, and that also published, and you can read more of that in this paper. And with that, I would like to also thank the funding resources, and thank you very much for your attention.
Sorry, Dr. Erdemci Tandogan, can you hear me?
Now I can hear you.
Okay. Thank you, Sinsley, for this enlightening presentation. And if you wish, we may move on to the our question answer season, which consists of the questions posed by our team members and the YouTube listeners.
That sounds great.
So shall we start?
That sounds great. Yes. Thank you.
Okay. The first question is I'm going to ask about considering the impact of the heat stress combination with genetic risk, LS, on the developmental process. Could a model be proposed in which it affects developmental pathway and leads to phenotypic outcomes?
Like development pathways and leads to phenotypic outcomes. Yes. We mainly study the mechanical aspect of the study. So there are, of course, molecular contributions coming from that, and then the genetics also changing, and as another factor. But this is one piece of the puzzle, and we do actually show that using this type of models and modeling these type of processes, we can explain some of the phenotypes that we see in the experiments in the system. So it is, in short, the answer is yes and no. The mechanical forces contribute, and it is part of the puzzle, and it also helps us to explain some of the phenotypes that we observe during embryonic development, for example.
Thank you for your answer. Another question is, I'm going to ask from our YouTube listeners. To what extent do you think temperature-dependent transcriptional regulation in Drosophila embryos influenced by the epigenetic factors, such as chromatin accessibility or nuclear organization?
Yeah, that's a great question. The temperature is definitely an important factor here. Indeed, if we perform the experiments at different temperature, we can get different outcomes. And from a physicist's perspective, the role of temperature here, that it changes the forces in the system as well. So it can actually make cells, like for me, the temperature in my simulations is this affecting the fluctuations of these cells. So from the mechanical perspective, yes, I can see that how temperature is important and impact. And that's my answer from a physicist's perspective. So that's definitely true. There's a temperature effect. But when we do the experiments, and when we do our control experiments, or when we change the repeat experiments with mutants, we make sure that we actually do at the fixed temperature, whatever the temperature we do, and we even do in similar time frames so that the environment is even similar. So we try to reduce those impacts in these type of models and then the experiments to support our hypotheses.
Thank you for your valuable response. I'm going to move on to another question. This is also from our YouTube listeners. How can computational evolutionary biology approach be utilized to understand the extent to which in hierarchical organization genetic expression networks in Drosophila embryos in conserved across species?
Oh, okay. That's a great question, which I'm not an expert in evolutionary perspective of these. So if I answer this question, it will be not good all the literature that I don't know. So from the computational, there's of course computational evolutionary area as well. But from our perspective, what we study from the evolutionary perspective only that I can comment on that, the models, the morphogenetic processes that we model for these embryos, for example, body axis elongation, it is a process that is true from fruit fly to zebrafish and to human embryo, right? So it is a process that is conserved. So that's much like we do computational across species that I can say.
Okay, and let's move on to another question. The next question is about the mechanism of strip information primarily regulated by transcription factors. However, how the epigenetic factors such as long-range chromatin interactions and enhancer promoter looping contribute to segmentation patterns?
Yeah, okay, that's another specific question relating to the genetic factors here. We are interested in the mechanical perspective of these things. So for us, when some of the experiments are done and knowing that these are modified in the experiments, what we look at to understand, are there some physical changes in the system? So we are looking from the perspective that if these are changing some conformations, some such as cell shapes, for example, or some conformations of these proteins, for example, how they move and so on, that's the piece that we can insert this type of mathematical models. And that's what we model.
Thank you again. Let's move to another question. How do cells develop resistance mechanisms to temperature fluctuations? Are these processes deterministic or stochastic in nature?
Okay, could you please repeat the question? Okay, now I see the question. How resistance mechanism to temperature fluctuations, are these processes deterministic or stochastic in nature? As far as I know, there's like a lot of stochasticity in these systems, and the cells are, in terms of resistance to mechanisms, the cells are embryonic development are very robust. So, and that's another thing, actually, it's very tricky to modify these in the experiments, but in mathematical models, for us, these are some parameters, right? We turn it on or off. But in the experiments, that's not the case. Embryos are very robust and they are resistant to changes. And that's why it is hard to study these in vivo in the actual systems. And so that's I guess, I hope this answer your question, is that yes, they are resistant and they are very resilient. That's why we get very robust embryonic development. And in terms of the stochasticity, there are lots of stochastic fluctuations, for example, happening for the cells and during embryonic development. And that's why we get, like, when we do these experiments, that's why we get such huge error bars, for example.
Thank you for your response. Another question is, to what extent do organism scale models accurately reflect molecular scale information? At what point should multi-scale modeling approach be integrated?
Yeah, so the question is that at what extent organism scale models accurately reflect the molecular scale information, and what point should multi-scaling modeling approaches to be integrated? So that's another great question. These are happening at different scales, of course. There's the contribution coming from the molecular scale and at the multi-scale. But as a physicist, what we are interested in understanding, actually, without going too much in details, for us, the coarse-grained simulations is what we want to do. So in other words, like for us, a cell is an elastic polygon. So without going too much into what is happening in the cell in the molecular level, we are interested in understanding, with that simplification, can we explain some of these questions? And the answer is, so far, getting the experimental match between the model and the experiments, it always amazes me. With these simple physical models, we are indeed able to explain these morphogenetic processes, for example, without going too much into detail of how like molecular scales that we would introduce them. In other words, like we do not go into details almost in purpose, right? We want to keep our models simple to be able to explain the simple physical mechanisms behind them.
Let's move on to another question. To what extent do organism scale models? Another questions coming from our YouTube listeners. I'm confused a little bit about. I'm sorry for that. What experimental approach are utilized to measure T1 transition timescales in vivo, and how do these measurements correlate with observed changes in tissue material properties?
Yes, they do. There are some data, not so much, but there are some data measuring the T1 transitions for fruit fly embryo, again, during germ extension, for example. And they do study, for example, some specific role of tricellular junctional proteins contributions when they modify compared to wild type versus the mutant embryo. They repeat the analysis and they see that actually from mutant to mutant, T1 time, transition time is different. And they do basically measure that by imaging the cells and annotating and looking at where the cell vertices are and tracking them over time. So time-lapse movies of the tissues, and with a membrane marker that you can observe the cell vertices over time, they are able to measure it.
Thank you for your response. Again, another question is coming from our YouTube listeners. Considering the role of stochasticity and natural networks in biological systems, how can the gap between mathematical models and biological reality can be bridged?
Yeah, the neural networks, even though there is it is not we do not do in my group neural networks and in that type of modeling, but there are definitely people started using neural networks to study vertex model types of models. And the idea is that predicting where these cells are going to go in the next time step, right? So there is an opportunity, I think, to study that and to understand. But at the same time, these are systems with in very flexible or soft. So these are soft materials. That's why we call them fluid-like materials. And in that case, their stabilization is not easy. In other words, they can go from one state to another state very quickly and change the states. And that makes it difficult to develop neural networks type of models on them too. And that also makes it difficult to kind of do energy minimization on these type of systems. But maybe their neural network type of models will help them. I don't know. People just started working on them. And that might help the gap between the mathematical models and biology. If the gap here is referred to how accurately we are modeling them, that might further help. Yeah, that's my answer to that. Yeah.
Thank you for your response again. And I'm also going to continue with another question. How do variations in teratogenic effects observed across different animal models align with human data? How can these discrepancies be resolved?
Okay, I'm trying to see the question from my end as well, but I do not see it. Sorry. Okay, it doesn't show up on my end again. So if you could repeat that, yes, please.
Okay, we can move another question if you want.
Oh, you can. If you can repeat the question, I couldn't hear it, and I'm trying to read myself.
It's okay. I can repeat it. How do variations in teratogenic effects observed across different animal models align with human data? How can these discrepancies be resolved?
Yeah, I never work with human data, or I never compare even zebrafish data with fly data and so on. From the modeling, from the physicist's perspective, for us, these cells, for example, change shape and migrate, right? And if something is changing shape, there are physical forces involved, and we're interested in understanding that physics behind of them. And from that perspective, I do not see differences between, for example, these forces forming during morphogenesis in fly embryo versus zebrafish embryo versus human embryo. From species to species, from our perspective, in terms of physical forces, we do not think about the differences from that perspective. That answers your question.
Thank you for your response. I'm going to ask the last question, and it will be conclude the final question for you. The question is, your findings, yes, as I said, your finding indicated that T1 transition dynamics influence tissue viscoelasticity. Do you think T1 transitions could serve as a predictive factors for tissue level stress relaxation behavior in developmental process.
Yes, so the proposal of the model, like what we propose at the end, during developmental processes, organisms might be using this as a process to change the global tissue response, which viscoelasticity, for example, is one of the tissue global response. So this is not verified, this is not tested in the experiments, so it's harder to answer that question without verification, but that's what the model predicts and proposes.
Yes. Thank you for your valuable answer. Dr. Erdemci Tandogan, thank you very much for taking your time to share your knowledge with us and for your informative presentation. And on behalf of myself, our team, and all the participants, we have to say thank you again.
Thank you very much.
Thank you. We wish you all the best for your health and your success in your time and your studies. Thank you.